Hello everyone, my name is Rasib Singh and today I'm going to present my presentation for assignment 103. So first of all, let me share my screen. So here's my screen. I'm gonna cover the major topic of biological activity, which is totally concerned and did by Sarah Smith or her team in 1998, and this presentation is going to submit to Professor Lee. So in this particular study I'm gonna discuss the stress on memory, which is based on humans, and rats, which is animals. So it is differentiate between two things, humans and animals. So to start with study, the human study is a line of cognitive psychology which generally focuses on the comprehensive mental functions, including memory, attention, and perception, including the human study. This study specifically looks at how stress affects memory function, which is a crucial component of cognitive functioning. This field of study is crucial comprehending how the stress, how the stress and the other environmental influences may affect human cognitive functions. Next, we gonna discuss some major outline of overview of the animal part, which in animal part, the study part of behavior, neuroscience investigates stress impact on memory performance in rats, focusing on neural mechanics causing cognitive impairments. Using animal models, researchers can study neutral pathways and neurotransmitter systems, providing insights into cognitive processes. So, moving forward, here's some human study and animal study, which is maybe comparison a little bit. And for start with human, the cognitive appearance theory suggests that individuals assess stressor based on perceived impact on well-being cognitive resources. The hypothesis is acute stress can impair memory or process in humans as cognitive resources are used to cope with stressors. And moving forward, the animal parts, the Glucocorticoid theory suggests that human-induced release of glucocorticoids can negatively impact hippocampal function and memory. And the study suggests the rats exposed to acute stress may experience impaired memory performance due to impact of glucocorticoids or hippocampal campbell function in the systematic plasticity. So now I'm gonna cover the major part, which is the experiment. So in this experiment, it's majorly the impact on the performance of humans. So as a subject, we take 60 undergraduate students aged between 18 to 25, right? And they are assigned randomly in groups. So the variable measured is that we will use the journal method to recognize all the memory task, which is body code and memory task for humans. And then stress indication techniques, which is for humans, is trial social stress test for humans. So now research design. In human study, randomized controlled trial RCT with two groups, test groups and control groups. And moving forward in data collection methods, humans participants completely the memory task before and after stress induction. Their performance scores were the compared to time points. And now the key finding and the conclusion. So for humans, the stress showed a significant decrease in memory performance compared to the control group, in which T58 equals to minus 2.34, which is really less. Generally, and the other one is P is 0.05. This suggests that acute stress impaired memory functions in humans. And now let's discuss about rats or animals. So here we took 40 male visitor rats aged between 8 to 10 weeks, and they also randomly assigned. So about the memory of performance access using maze tests, and then instructions maze are brain's maze for rats, stress induction techniques, which is social defeat for rats, and then an animal study is quasi-experimental design with two studies, stress group and control group. So in data collection method, rats underwent the maze task before and after the stress induction. Their performance time to complete the maze was compared to between time points. So the key finding conclusions of animals is the stress big group exhibited a long time to complete the maze compared to the control group, in which T38 equals to 3.25 and P is 0.01. So indicating impaired memory performance in stress rats. So in comparison of both methods, human study allows to investigate the impact on memory in a controlled environment, providing insights into human cognition and potential interventions for stress-related memory impairment. And secondly, for the animals, it enables researchers to study underlying uh, neuromechanisms of stress-induced memory impairment, opening insight into the biological basis of related cognitive dysfunctions. So moving forward, some advantages and some disadvantages. So here, I would like to discuss the advantages. In human study, the advantage is that the humans are really complex to study, and it helps to such as we can encode memory easily, and we can investigate high and complex memories, and plus provide in human study provides insight into effect of stress or exercise on the human behavior and cognitive in real world. And moving forward, the limitations. As we can see, the ethical constraints may limit the type, invention, or manipulation because while in the study, some human can be manipulated or can be do manipulations. So because of that, the result would be not correct. And plus, the limitation is that human studies may suspicious to co-founding variables such as lifestyles and the individual factor designs, or we can say environment, because environmental change is really affect their behavior. And moving forward in animal section, animal allows investigation to underlying biological mechanisms that may not be crucial to human studies, as we discussed before. And plus provides control over environmental factor genetic background, reducing variability in results. So in the limitations, we can see that finding animal models may not translate directly to human due to the different brain structure, as well as ethical consideration regarding the use of animal research. So in the last, I'm going to discuss the most efficient method in different circumstances. So for human subjects are ideal for the study unique aspects of cognitive behavior or physiology, complex social or psychological processes in animals and the effects of interventions or treatment specific to humans, such as psychotherapy or paramedic, uh, param, uh, paramedical treatments. And for the animals perspective, as we can see, animal studies are ideal for studying considered biological process like neuromechanism, sensory process, and living and memory. They are also valuable for investigating the effects of interventions or treatment that are not visual or ethical to study in humans. So here are the references. That's from authors. Thank you so much.